Don't applaud early. I will go ahead and get started. Welcome everyone, and thank you all for being here today. As you know, Alexis is defending her PhD today. So that's going to involve a 45 to 50-minute presentation of some of the data that she's collected in the last few years. And we're all excited to hear about her progress and the importance of her work. We will have an open Q&A session after her presentation, and I invite everyone to please ask those burning questions that you need for clarification or to understand why in the world she took this on in the first place. I'm particularly excited to be able to introduce her today because Alexis has just been a fantastic member of our team. She came into the lab and took on a challenging problem related to mechanisms of nicotine withdrawal and working on behavioral effects of nicotine or any other addictive drug in animal models is particularly challenging. And she was fearless in taking on really difficult experiments and really generated some exciting results that we'll hear about. In the meantime, every laboratory, every human endeavor has all sorts of turmoil that can occur. And when those events happened in my lab, Alexis was the calm, reasonable, sort of level-headed touchstone for me. Alexis, are we falling apart? No. We're doing okay. Anyway, it will be fantastic to see her move on and continue with her medical training. And I know that she's on her way to being a fantastic physician scientist in the future. And we are going to miss her terribly, but it's very exciting to have you here presenting the role of the lateral dorsal nucleus in nicotine withdrawal.
Hey, thank you, Dan, for that wonderful introduction, and thank all of you guys for being here, friends, family, and colleagues. I have some family here all the way from California, so I'm just incredibly grateful for everyone's presence here today. And I'm very excited to tell you about the work of my thesis project, which is titled The Role of the Lateral Dorsal Tegmental Nucleus in Nicotine Withdrawal. And so I want to start off with a little outline of what I'm going to be talking about today. So I want to really introduce the background material that was the basis for my thesis project. And so we're going to start with talking about the current state of nicotine addiction. And then we're going to follow it up by defining some of the key brain circuits that are underlying my project. And then finally, we're going to get into the real meat of my thesis, which is the results section. And that's going to include talking about nicotine withdrawal symptoms. And this is going to be more high-level behavioral data. And then we're going to zoom in on the brain a little bit and talk about changes in brain activity and dopamine release. And then finally, we're going to close out by zooming in even farther and dissecting the circuit anatomy. So let's start with the current state of nicotine addiction. So the tobacco industry has a market value of hundreds of billions of dollars, and the tobacco plant is cultivated from many regions across the world. And when they develop tobacco products, they utilize different primings or ripenesses of this tobacco plant to really curate a product that's marketable to the general public. And all of these products have nicotine in them, which is the main psychoactive stimulant. And we find it in things that we traditionally think about when we think about nicotine, like cigarettes. However, it's also present in things like hookahs, pipes, cigars, smokeless tobacco, and e-cigarettes, which have greatly gained popularity since they hit the market in 2007. So we can see that nicotine substance abuse remains a major public health problem. Currently, tobacco use disorder is the leading cause of preventable disease, disability, and death in the United States. And there is still a very high number of cigarette users, so about 30 million in the US alone. And nicotine is a really unique drug of abuse in that it has a very high transition to dependence. So about 2/3 of smokers will transition to dependence in their lifetime. And the health consequences surrounding nicotine and tobacco use are very widely known, with about 16 million people living with tobacco-related diseases in the US. And these diseases can include anything, including cancer, heart disease, and lung disease. And so because these health consequences are very widely known, there's a large proportion of smokers who wish to quit. However, it remains very difficult to do so. So it's estimated in some studies that it takes about 30 quit attempts to remain abstinent for just 1 year. And so it's really important for us to think about how nicotine addiction is happening and how we can help people successfully quit. So nicotine itself is very highly addicting, and this positive reinforcement leads to continued nicotine seeking. And after this happens over a repeated amount of time, there happens to be changes in the brain that will lead to a withdrawal state upon cessation from nicotine use. And it's really a combination of this positive and negative reinforcement that lead to continued nicotine seeking. And during periods of abstinence, this leads to relapse. And so this happens in not just nicotine addiction, but a lot of drugs of abuse. And we've even seen evidence for it represented in the media. So if we take one example, Captain Jack Sparrow, we know that he, throughout the franchise, consumes a large amount of alcohol, specifically rum. And it's very clear that he's dependent on it because he gets very agitated. Why is the rum gone? Throughout the entire series, which means that he has this continued seeking and craving of the drug, eventually leading to relapse. And so if we don't want to maroon people on a deserted island, how can we actually help people quit? So luckily we can develop different treatments, right? So if we start at the beginning of the road with our health problem, that is tobacco use disorder, and we want to end with a treatment. So I'm going to be using one of the gold standards for nicotine cessation, which is varenicline. And it took a series of preclinical and clinical steps to get to this point. So first, you really have to understand what the neural mechanisms of nicotine's action in the brain are. And this will allow you to identify certain drug targets. So for varenicline, that's going to be the alpha-4 beta-2 nicotinic acetylcholine receptor. And varenicline was derived from a molecule that was a partial agonist of this receptor and was thought to have moderate effects on smoking abstinence. And so they took this drug and they tested it in preclinical models to see how it would work on the dopamine systems and circuits that are really important in nicotine addiction. They also tested it in behavioral models. So what does this drug have effect on nicotine intake, reward symptoms, and withdrawal symptoms? And only after it successfully moved through all these preclinical steps could we take it to clinical trials and see if it's efficacious in humans. Indeed it was, and it eventually went on to become FDA approved, and we now prescribe it for people who are trying to quit smoking. And it's really great that we have this drug, but we are only able to boost abstinence rates to about 18 to 25% at 6 months. And so while this is really great that we're helping a portion of people successfully quit, there's clearly a lot of work that still needs to be done. And so this requires us to go back to the beginning of the road and really look at the neural mechanisms undergoing nicotine addiction and withdrawal, which is where my project lies. And so what are those neural circuits that I'm talking about? So nicotine acts in the brain through nicotinic acetylcholine receptors. So this molecule is going to come, bind to this receptor, and cause cellular changes. And we know that these receptors are expressed more highly in some brain areas over others. So a specific circuit that has a very high level of these receptors is the habenula and its projections to the interpeduncular nucleus. And it's really great that this pathway is heavily conserved from humans to mice, and mice are the model organism that I'm using for my studies. And so not only does this area have the highest densities of nicotinic acetylcholine receptors in the mammalian brain, but also the largest variety of subunits. And so to create a functional receptor, you have 5 of these subunits that come together, and we designate them as either alpha or beta subunits. And it's actually been shown that in humans, nicotine addiction in humans, that specific nicotinic acetylcholine receptor genes, mutations within them are a risk factor for smoking. Specifically the alpha 5, alpha 3, and beta 4 gene cluster. And this is really interesting to us because it's these same exact subunit types that are expressed very highly in the medial habenula and the IPN. So altogether, what I've told you so far makes the IPN a really interesting target to study nicotine addiction and withdrawal. And in fact, we're not the first lab to do this. So a lot of labs have looked at the IPN, which is composed largely of inhibitory GABAergic neurons. And it's been shown that increased activation of these GABAergic neurons and excitatory drive from the habenula, specifically the medial portion of the habenula, are important in driving nicotine withdrawal. And other labs have shown that optogenetic manipulation of the IPN in nicotine-dependent animals can actually alter the withdrawal symptoms. And so this led to my project that I really wanted to study the IPN in withdrawal, but I was actually very interested in what the downstream targets of the IPN were. And so one of those connections is the lateral dorsal tegmental nucleus, or LDTG. And a big motivation behind my project actually stems from Shannon's work in the lab. So she was a grad student in Dan's lab before me, and she was really interested in the LDTG because it's been known to be a major regulator of dopamine release. So it's part of the reward system, and it drives excitation of midbrain dopamine neurons through the various cellular types within the LDTG. And the reason that we care about dopamine is because during nicotine addiction and withdrawal, it's been shown that dopamine neuron activity and dopamine release is actually depressed. And so in Shannon's work, she was also interested in the connections from the IPN to the LDTG, but she was looking at different doses of nicotine. So at lower doses of nicotine, you get very rewarding effects. However, as you increase the amount of nicotine intake, it actually becomes very aversive at high doses of nicotine. And this is a reason why smokers titrate their intake, right? So they only smoke certain amounts because they don't want to get to these high aversive levels. And so in Shannon's work, she looked at the connection between the IPN and the LDTG and found that it was a dense functional GABAergic projection. So if she optogenetically manipulated this circuit, she saw inhibitory currents that were blocked by bicuculin, which is a GABA receptor antagonist. And what she saw was that if she blocks this circuit, so she inhibits this circuit during high dose nicotine aversion, you can see that in our control animals, anything that's below 0 on this preference score indicates aversion. And if she inhibits this circuit, she actually stops that aversive response from happening. And so this really shows us that the IPN to LDTG might be really important in mediating aversion. And so for my project, I wanted to ask the thesis question of how this pathway mediates the aversive nicotine withdrawal state. So let's get into actually some of the data that I found. So first, we're going to talk about the nicotine withdrawal symptoms. So to study nicotine withdrawal in mice, we have to expose them to nicotine. And to do that, we're putting it in their drinking water. So these mice are exposed to a nicotine tartrate solution that we administer in their drinking water. And we combine it to tartrate because it's an easily dissolvable substance. And so that means that for our control animals, we give them a tartrate solution to control for that compound. And so this gives us several experimental groups. So our nicotine naive group is exposed to tartrate in their control water for 4 weeks, and they never experience nicotine. Our nicotine dependent group received 4 weeks of forced nicotine tartrate exposure. And what we see is after 4 weeks, we're able to monitor the body weight throughout this experiment. And we see that these animals are maintaining a healthy body weight control compared to their control counterparts. We do see a slight decrease in their body weight compared to the controls at the end of our experiment, and this is likely due to the fact that nicotine is an appetite suppressant. However, they maintain a healthy, normal body weight. We also see that after 4 weeks that they have elevated serum cotinine levels. And so cotinine is the major metabolite of nicotine. So high levels of cotinine indicate high levels of circulating nicotine. And then if we put them through 24 hours of withdrawal, so we remove nicotine from their drinking solution and replace it with their control solution, you can see that those serum cotinine levels are depressed.
Can we ask a question, or should we say?
You can ask a question.
What is the average?
Huh?
In terms of grams.
In terms of grams, yeah. So we're using 8-week-old mice. So just as they reach adulthood, so males and females will be a little bit different between the two. I don't know exactly what the average weight, but normal 8-week. Yeah, and we would, if we saw any body weight reduction greater than 20%, we would exclude them from the experiment, but we didn't see that effect. So yeah, this is called spontaneous withdrawal, is when we remove the nicotine from the drinking solution and replace it with the control water for 24 hours. However, there's a second method of nicotine withdrawal induction, and that's injection of mecamylamine, which is a nicotinic acetylcholine receptor antagonist. And it's been shown in the literature since the 1990s that this precipitates a withdrawal state. And so within our different experimental groups, our naive group will receive an injection of mecamylamine to control for the effect of the drug alone. Our dependent animals will receive a saline injection, so they will not be undergoing withdrawal. And then our withdrawn group will receive an injection of mecamylamine to precipitate a withdrawal state. And I want to give a huge shout out to Nicole, who really trained me on all of these different nicotine methods. So what does nicotine withdrawal actually look like? So it's a combination of both somatic and affective components. So somatic components are the physical manifestations of withdrawal. And in humans, this might look like tremors, itching, or GI discomfort. And in mice, this will look like grooming, paw licking, or shaking. And then the affective components are the more emotional components of withdrawal, so anxiety or depression. And in mice, this might look like decreased exploration or interaction with normally rewarding stimuli. And so here's a couple quick videos to show you what these differences might look like. So here's an animal in its normal baseline state. And what you can see is this animal is exploring, exhibiting normal exploration. So it's sniffing around, it's using the entirety of the chamber. This is just considered normal mouse exploratory behavior. But if we take this same mouse and now we're putting it through nicotine withdrawal, you can see that it's exhibiting very different behavior. So this mouse is going to stay in the periphery of the box, so it's not exploring the center as much. It's also going to spend a lot of time tending to itself. So grooming, paw licking, shaking, those kind of behaviors. And this is very indicative of a withdrawal state. And so now that we have a way to actually study nicotine withdrawal, I wanted to know how these withdrawal behaviors change after inhibition of the IP and LDTG circuit. So we're going to be blocking this connection and see how it changed withdrawal behaviors. So to block this circuit, we're using optogenetic inhibition, and I have to give a huge shout out to Ross, who helped me establish this method in the lab and get proof of concept data in wild-type mice. And so in this method, we're going to be injecting either a Cre-dependent halorhodopsin, which is our inhibitory opsin, or a Cre-dependent control virus, which is EGFP, and means that we're not inhibiting this circuit. So we'll be injecting that into the IPN of GAD-Cre mice, and all this means is that we're restricting expression within the GABAergic neurons, which is the population that we're interested in. We'll then be placing a fiber above the LDTG for optogenetic inhibition of the terminals. And so you can see expression in the IPN in these histology images. So in the blue, we're just showing DAPI, which is the nuclei of the cells. And then if we zoom in on the IPN, you can see in green is the expression of our Halorhodopsin inhibitory virus. If we do histology within the LDTG, you can see the fiber placement above the LDTG. And if you zoom in, you can see expression of the virus in the terminals. And so this looks very similar to the videos that I showed you before, except this time the mice are now hooked up to a fiber optic for light delivery to the LDTG. So you can see that these mice are allowed to exhibit normal behavior. They can freely move around, but under these circumstances, we can now manipulate the circuit and shut down the IPN to the LDTG. And so you can see that we have functional expression of this receptor within GABAergic neurons of the IPN. So we stained for glutamate decarboxylase, which is labeled as GAD here, and that's a marker for GABAergic neurons. And you can see in orange, there's overlap between the GABAergic population and the population that is expressing our Halorhodopsin. If we do ex vivo electrophysiology, we can see that we get light-evoked hyperpolarization and light-evoked inhibitory currents. So now that we have a method of inducing nicotine withdrawal and a method of blocking our circuit, we can use it in a variety of behaviors. So this is a timeline for the first experiment that I'm going to show you. We begin them on nicotine drinking on week 1, and we do surgery at the end of week 1. We then go on to habituate the animals to injections, and then we're going to be placing them in a real-time place preference box. So in this box, the mouse has the ability to explore 2 neutral chambers. These chambers have different patterned walls and different textured floors, so the mice can distinguish between the 2 chambers. And you can see that on the first day, we're just going to allow them to explore both of the chambers with the light off. So we're just gauging baseline preference. And then on day 2, we're going to turn on the light with one chamber and not the other. And so here you can see these are some position traces of the animal. So over here, these lines are representing the path of the animal throughout the experiment. And what you can see is that in both our naive and our withdrawn groups, these animals on the pretest day are spending equal time in both chambers. Then if we look at the day where we inject mecamylamine, you can see that our naive group doesn't care about the light being turned on. However, our withdrawn group is spending a high amount of their time in the light-paired chamber. And you can see that represented in these graphs here, where our naive and dependent groups don't care, especially if we look here with the Halorhodopsin group, which again is the group receiving optogenetic inhibition. The naive and the dependent groups don't care about the light being turned on, but in our withdrawn group, all the animals develop a preference for the light-paired chamber. And so this really suggests to us that blocking this circuit is alleviating the aversive nicotine withdrawal state. So then I continued these experiments across 3 days of conditioning. So they're receiving the same procedures on days 2, 3, and 4. And then on day 5, we have a post-test day where we turn the light off. And now we're gauging whether, because they've had this pre-exposure to this light-paired chamber during withdrawal, do they now care on the post-test day what chamber was previously light-paired. And so you can see here that across our 3 days of conditioning, they maintain preference for the light-paired chamber. And then we see something interesting on the post-test day, that if they're in their baseline state, they don't really care about which chamber was previously light-paired. However, if we put them back in a withdrawal state, they are now seeking out the chamber that previously had the optogenetic inhibition. So again, all of this really suggests to us that blocking this circuit is alleviating some of this nicotine withdrawal state. And so the next question that I wanted to ask is, what specific behaviors are being modulated by this circuit? So the first thing I looked at were the somatic signs, which again are those physical manifestations of withdrawal. And again, I have to shout out to Aaliyah, Ross, and John, who all helped determine how to score these videos. in a blind and independent manner. And Sophia Bennett is the one that used other people's training data to learn how to score them and applied it to this data set. So what you can see here is if we just combine all the somatic signs together and look at the duration of time spent performing these signs, that during nicotine withdrawal, we have elevation of the time spent performing these somatic behaviors. So they're spending more time tending to themselves. And then with our optogenetic inhibition, we're able to bring that duration back to levels of naive controls. If we break this up by individual somatic sign, we can see that paw licking and shaking are the most indicative of a withdrawal state. So that's the somatic signs involved in nicotine withdrawal, but I also wanted to see if this circuit was involved in the affective behaviors. And so we can't really ask mice if they're feeling anxious, so we have to use different tests to kind of gauge their anxiety state. And so one of those tests that's been used in the literature is called the open field test. And in this test, they're placed in a large chamber box, and under normal conditions, these mice will explore the entirety of the chamber. However, during nicotine withdrawal, you can see that the path of this animal is showing us that they're not spending as much time in the center zone. But if we inhibit our circuit, you can see that this withdrawn animal looks very similar to our naive controls. And so we see that reflected in that the number of center visits is depressed during nicotine withdrawal, but is then recovered with our inhibition. And the same is true for distance traveled. So again, this suggests to us that we are alleviating the anxiety-like state of the animal. We didn't want to use just one effective test to look at behavior because mouse behavior can be very variable. So we tested this across a couple of different tests to look for this effective behavior. And so the next test that we looked at was novel object interaction. So this is very similar to the last one, except this time we are placing a novel object in the center, which is a Lego. And you can see that the time spent interacting with this novel object is decreased in nicotine withdrawal and then recovered with the optogenetic inhibition. So this is more evidence pointing towards this circuit being very important in the effective withdrawal state. And the last test I did was a test of anhedonia. And anhedonia is the reduced ability to experience pleasure or reward. And this is often a symptom of depression. And so to test this in mice, I gave them the option between sniffing a swab that's dipped in water and sniffing a swab that's dipped in the urine of a mouse of the opposite sex. Under baseline conditions, so you can see in our naive control groups, they'll spend more time sniffing the urine swab over the water swab. However, during nicotine withdrawal, you can see that they are exhibiting an anhedonia state where they are not having a preference for one swab over the other. However, with optogenetic inhibition, we're able to recover that back towards the baseline hedonic preference. And so let's kind of summarize what we've gone over so far, right? So if we zoom in on this IP into LDTG circuit, you can see that we have increased activity of the GABAergic neurons, presumably releasing GABA onto the LDTG. And we've shown that this connection is important in both the somatic and the affective components of nicotine withdrawal, and that inhibition of this circuit is preferred during a withdrawal state and alleviates these nicotine withdrawal signs. And so I want to give a little bit of motivation behind the next part of my talk. So we know that this is important in nicotine withdrawal, but I'm also interested in what's happening downstream to dopamine release. So we know that the LDTG is regulating dopamine release through dopamine neurons in the VTA. And so this next part is really going to be looking at what's the activity of the LDTG like during nicotine withdrawal and what is the corresponding dopamine release. So this is where we're going to be looking at changes in brain activity and dopamine release. So we're going to be switching up the methods a little bit here. So this time we're going to be using GCamp to measure calcium changes in the LDTG. And an increase in calcium is indicative of increased cellular activity. And so you can see expression of our GCAMP molecule here, and we're using fiber photometry to record these changes in fluorescence. And so you can see this that the LDTG is spontaneously active, and these transients are indicative of changes in calcium release or calcium in the cell. And so what I wanted to do is measure the LDTG activity over time. And I wanted to measure it before and after an injection of saline or mecamylamine. So we can compare the baseline state before injection to the post-injection period. And so what we see, this is a nicotine-dependent animal, and if we inject them with saline, you can see that it's spontaneously active before the saline injection and after the saline injection. However, if we inject them with mecamylamine to induce withdrawal, you can see that it's still spontaneously active, but it seems to be that the magnitude of these responses are decreased. And that becomes clear when we average our data across multiple animals, that there's no change in our tartrate nicotine naive group. However, our nicotine dependent group has decreases in LDTG activity during nicotine withdrawal. And this isn't necessarily surprising since we are assuming that there's increased GABAergic drive from the IPN during nicotine withdrawal. So to further quantify this, we looked at a couple of different peak attributes. So we counted up the number of significant peaks that we saw in the baseline period compared to the post-injection period, as well as area under the curve within a specific time window, and the peak amplitude. And so what we saw is that we didn't have a change in the overall number of peaks. However, in nicotine withdrawal, we saw a decrease in the area under the curve, as well as the peak amplitude. And so all of this again suggests that during nicotine withdrawal, there is decreased LDTG activity. So I wanted to compare what was happening in the LDTG to dopamine release within the accumbens. So to do that, we're using a very similar method, but instead of GCAMP, we're putting GrabDA within the nucleus accumbens, and this is a dopamine sensor. So when dopamine is released in the nucleus accumbens, it will bind to these receptors and we'll get changes in fluorescence. So you can see the virus being expressed within the nucleus accumbens, as well as spontaneous dopamine transients. And so then again, if we compare dopamine release before and after a saline injection versus before and after a mecamylamine injection, you can see that we get very similar results, that we are getting depression of dopamine release during nicotine withdrawal. And so this really fits together nicely with our story that the IPN is shutting down the LDTG, which is in turn decreasing dopamine release. And so again, we looked at the peak attributes of these dopamine transients, and you can see there's no change in the number of peaks, but there is a decrease in area under the curve and peak amplitude. And so this was very interesting, but I did get a pretty cool result while I was exploring the LDTG with these methods. And what I saw is that the LDTG has a very big response to novel object interaction. So we're going to watch the calcium activity of this mouse as it approaches a novel object. And so you can see as the mouse approaches this object, we get an increase in calcium activity within the LDTG. And so this was really cool because it gave me a way to probe the system and look how changes to novel object interaction change before and after withdrawal. And so we can see that in the LDTG, if we present a novel object on day 1, we get increase in LDTG activity. And if we give a second different novel object on day 2, we're able to still elicit that same response. So this is a really strong technique to be able to measure this LDTG response before a saline injection and then after a mecamylamine injection to precipitate withdrawal. And so you can see an example trace of an animal here where all of these red lines are novel object approaches. And so you can see the majority of the time there's an increase to novel object interaction. And then in a small percentage of cases, we have no response to novel object. And so here's a heat map of that same animal after a saline injection. So you can see this white line here is when they approach the novel object, and these warmer colors indicate more activity. So it's highly active after novel object interaction, but then when we put it in a withdrawal state, we no longer see that same activity. So if we compare this to our tartrate control group, you can see that before and after saline or methylmalonine injections, they're still responding to novel objects. However, during nicotine withdrawal, we don't get this same increase in response. And so if we look at the percentage and we compare the amount of increased activity to novel objects interactions that had no response, you can see that across our control groups, we have a pretty high level of novel object interactions that cause increased activity. However, during nicotine withdrawal, which is this final pie chart, you can see that this number of increased activity, novel object interactions, is decreased. And so we wanted to see then, again, what is happening to dopamine during the same kind of test. So interestingly, we do see novel object interaction induces dopamine release within the accumbens. And we see these kind of two similar responses of either there's an increase in dopamine or no response. And it's very consistent with the LDTG that this is the kind of response that we see in LDTG activity, and dopamine release has this same kind of response to novel object, and the percentage of novel object responses that cause this kind of activity is decreased in nicotine withdrawal. Where it gets a little bit different is that if we look at these no interaction responses, or these no dopamine release to novel object interaction, you can see that nicotine withdrawal doesn't increase this number as we would expect from our LDTG data. And the reason for that is because we actually see more than these two different responses. So in a subset of cases, we do see an increase in dopamine right before the novel object interaction, which we did not observe in the LDTG. And so we can actually break down this dopamine release into a couple of different categories. So we have this post response, which is very similar to our LDTG response. We have the no response, we have a pre-response, and then a very, very small percentage of the time, we actually saw this elevated level of dopamine that decreased with novel object interaction. And so if we compare our percentages again, what we see is what I pointed out before, that we have a decrease in this number of post responses, but it's not filled in by more none responses. Instead, we have an increase in this pre response. And there is some precedent in the literature that anticipation of novel reward can increase dopamine release within the VTA. So what we are seeing here could be an anticipation response, but we're not exactly sure what it is yet. So we need to modify our summary a little bit. So now we're including the IPN to LDTG to this dopamine system. And what we see is a decreased LDTG activity during nicotine withdrawal and decreased dopamine release within the accumbens. And so when it comes to novel object interactions, we see a decrease in the LDTG novel object response, but we see a kind of shift in the dopamine release to novel object. And so to kind of finish out this story, I was really interested in the heterogeneous cell types within the LDTG. So within the LDTG, we have cholinergic, GABAergic, and glutamatergic neurons. And I was really interested in which of these cell types is the IPN projecting to and might be involved in the nicotine withdrawal state. So this is going to bring us to our final section of results for dissecting the circuit anatomy. And so to do this, we're going to be using rabies monosynaptic tracing. And I want to give a shout out to Lauren, who really helped me get this new technique up and running in our lab. But essentially what we're going to be doing is we're going to be getting rabies virus within the specific cell types in the LDTG. And then we can back label our IPN inputs. So this is a virus that moves retrogradely or backwards. And so we can start in the LDTG and back label our IPN inputs. We can then use a secondary stain to stain these GABAergic IPN neurons for GAD, which is again a marker for GABAergic neurons, and see if these back-labeled populations are GABAergic, which is the cell type that we know is important in withdrawal. And so to do this, we are using a G-deleted rabies virus. So this is a modified rabies virus that if we just put it in the brain, it's not going to be able to infect any cells, and it's not going to be able to back-label any cells. So to successfully do that, we introduce a helper virus that's going to allow the rabies virus to do those things. But we only express the helper virus within one of these 3 cell types. And so here's an example of what this looks like within the LDTG. So you can see we have a couple of different populations of cells. We have these green cells, which are helper virus alone, which means they took up the helper virus, but the rabies virus did not infect them. We have these merged populations, which have green and red, which are suggestive, which indicate our starter cells. And that means that they had the helper virus and successfully took up the rabies virus for that back labeling. We see evidence of local connection back labeling within the LDTG in these rabies virus cells alone. So these are the cells that are projecting to our starter cells, but in a local circuit within the LDTG. And if we look at where this helper virus is expressing, we see that we are getting cell-specific labeling. So our helper virus should only be expressed in GABAergic neurons because we're using a GAD-CRE mouse. And so if we stain for GAD, we see nice overlap between those 2 populations. However, if we do the same thing in a V-GLUT mouse, which the helper virus should only be expressed in glutamatergic neurons, you can see that it is a distinct population from that GAD stain. And so if we get into the numbers, we can compare, how many starter cells are we getting within the LDTG and how many local connections. So right now, we're ignoring the IPN for this slide and just looking within the LDTG. And you can see that across our different cell types, we're getting about the same number of starter cells. However, there's different number of local connections with more happening to the GABAergic and glutamatergic population. And so finally, if we look into the IPN, you can see that we do get these nice back-labeled rabies neurons. And again, we're doing this within our 3 different cell types. And then we're going to stain the IPN to see if we are getting GAD negative or GAD positive populations. And what we see is that the IPN is projecting to all 3 cell types, but has less inputs to the cholinergic neurons and more inputs to the GABAergic and glutamatergic populations, which suggest those might be the important ones in nicotine withdrawal. And we can take this one step further and actually look at what subregions within the IPN these IPN or that these back-labeled neurons are coming from to each of our different LDTG subtypes. And the reason that I'm interested in these IPN subregions is that the ventral medial habenula projects to these subregions highlighted in green. And the ventral medial habenula is the part that's most implicated in driving nicotine withdrawal. And so if we look at our different populations, you can see that they're differentially expressed in these subregions. So the cholinergic neurons are mostly in the lateral section in the periphery of the IPN, where the GABAergic neurons are mostly in the central portion of the IPN, and the glutamatergic neurons are kind of expressed throughout. And so this brings us back to our summary schematic one more time. And what we can see is that we have all of our different behavioral outputs that are mediated by this IPN to LDTG connection. And now we can kind of break up our circuit based on the 3 cell types. So we have IPN projections to cholinergic, GABAergic, and glutamatergic populations, with an emphasis coming from lateral IPN to the cholinergic population, central IPN to the GABAergic population, and central and lateral to the glutamatergic population. And so that's the data that I'm going to be sharing with you today. I want to just bring it back to the summary one more time. There's been a lot of work done here, but there's clearly more work that can be done. And so a couple of future directions that we have is that it would be really great to specifically record the GCAMP signal within these three LDTG subtypes. So before we were just looking at LDTG as a whole, but if we break it down based on these subtypes, can we see differences in LDTG activity during withdrawal? And I think it would be especially interesting to look at the GABAergic and glutamatergic populations that receive a lot of IPN input. Additionally, we saw this really variable response within the nucleus accumbens, and I think future direction should be to figure out where exactly this dopamine signal is coming from. So it could come differentially from different cell types within the LDTG, but it could come from other regions as well, as the VTA dopamine neurons are controlled by many different areas. And with that, I will take any questions. Yep.
Very nice.
Thank you. Yeah.
One is IPN projection between we need some protein opportunity inhibition.
Mm-hmm. Yeah, that's a good question. So we are optogenetically inhibiting in our control animals, and we don't see the same effect that we see in our withdrawn animals. So naive versus withdrawn, we only see the effect in withdrawn. And we also did control for locomotor activity within our real-time place preference test, and we didn't see any changes in locomotion.
And then the second question is,
Mm-hmm.
there's no in the second novel activity.
Mm-hmm.
My question for you is that can you present the first novel because the increase?
Yeah. Yeah, that's a great question, and it's something that our lab has talked about a lot too. Is this no response interaction a product of becoming more familiar with the novel object? So that is definitely an option or an explanation for the data that we see. You can't control how many times the animal interacts with the novel object, so we don't know exactly when those novel objects are transferring from like a novel experience to a familiar one. And in a lot of the literature, they will do presentation of a novel object with a familiar object that it's already been habituated to and see the differences between the two. Unfortunately, because this is something that I just noticed while I was already running these experiments, I didn't run that, but it is an interesting question that we would like to explore in the future. Yeah. Uh-huh.
Is there a taste factor involved?
Yeah, that's a great question, and we do control for that. So nicotine has a very bitter taste and is actually not easily consumed by mice, and so we add saccharin, which is an artificial sweetener, and so that it's more palatable to the mice and that they'll drink it more. Another thing is we force them to drink this water, so they don't have an option to not, so they need the water for survival. And so, yeah, they will consume it and have enough levels to consume high levels of nicotine. But yeah, we use a sweetener to make it more palatable.
Okay, so that then they got control group as well?
Yep, in the control group as well. We give everyone the sweetener. Yep. Yeah. Oh, do I? I don't know. Thank you, Chandrika. Yeah, Josh. Yeah. So those are great questions. So nicotine does have peripheral effects, not just central nervous system effects. So it activates the sympathetic nervous system, and you do get increased, increased heart rate and those kind of like peripheral actions. So that definitely does happen. And then your second question was about mutations to the receptor. So yeah, there are mutations that are associated with higher risk factors for smoking, and they're specifically in the receptors found in the medial habenula and the IPN. Thank you. Yes. Yeah, so they're enhanced eating. Yeah, so no, I haven't tested changes in eating during withdrawal, but we do know from human studies that during withdrawal it does cause increased appetite. So I would assume that because we do see some evidence for decreased appetite on nicotine drinking water, that during withdrawal we would see that increased appetite, but it's not something that we have tested for. Yeah. Oh.
Go ahead.
Yeah, that's a good question. So people have done long studies. I've seen up to 72 hours. I can't recall any off the top of my head that have gone longer than that, but there might be. But actually what they've seen in mice is that across multiple days of withdrawal, the symptoms and dopamine depression get worse. I'm assuming that it will eventually return to baseline, but they have been exposed to nicotine for a long time, and those brain adaptations have already happened. Andrea.
That was fabulous.
Thank you.
I really liked your opening with that road to.
Yeah.
So I guess I anticipated that you were ended with here's the road to.
Yeah.
Human patients.
Yeah.
There's 30 million American smokers.
Yeah.
What does this help us? Even,
Yeah.
really leading to a medication, but what do you think?
Yeah, no, that's a great question. And we are definitely in the early phases of the preclinical steps that we don't have an exact drug target that we've tested or a drug that we're looking to see if it affects withdrawal. But it is interesting that the current drugs that we have for nicotine cessation target the receptors that are very highly expressed in areas like the VTA that control dopamine release, and the receptors that are very unique and uniquely expressed within the medial habenula and IPN have not been investigated as a potential drug target. So those alpha-3, alpha-5 receptors, and so that is a potential drug target that we could look at in the future because we know that it's heavily involved in withdrawal.
Why do you think that hasn't been studied?
That's a good question. I really don't know. I mean, the VTA nicotine withdrawal literature has been around much, much longer than this IPN literature has been. So maybe we're on our way there and just there yet. Yeah, still checking along. Okay. Yeah, Tess.
Okay, so this, I don't know much about my studies, so can I just get reassurance? The fiber surgery, like, is that distressing to the mice? Like,
Yeah.
Yeah.
Yeah, so they definitely undergo like a recovery period where they're more lethargic and stuff after surgery, but after they've recovered, they exhibit really normal mouse behavior that you would see in a mouse that didn't undergo surgery. The fibers that we use are designed to have very minimal damage to the actual brain. So the biggest inconvenience for them is that they have this head cap on them that's itchy sometimes. I mean, I can't ask them, but yeah. Okay. All right, I do have some acknowledgements that I would like to get through before you jump in, Dan. Okay, so first, I would like to thank the McGee Lab. You guys have made my experience in the lab so much better. Especially I have to thank Dan, my mentor, has just been so supportive to me throughout the 5 years here. I knew when I joined the lab that Dan was going to be a supportive mentor because even during my rotation project, He would be enthusiastic about the research, but he was also enthusiastic about me as a person. And what I didn't know to expect is that he was going to care about his students so much. So during COVID, Dan even let me borrow his car to get into lab. And even though the engine light did come on a few times, I don't think that most PIs would do that for their graduate student. So thank you, Dan. I also want to thank the McGee Lab has changed a lot throughout the years, but Ross and Siobhan have been constant during my time here. So I really want to thank them for their continued support. Me and Siobhan have kind of been like lockstep, hitting all the same milestones at the same time. So it's been great to have someone else to go through it with and make it feel slightly less isolating. And then Ross, you just have such an ability to connect with people immediately right off the bat. And we've had so many conversations that span, science to Marvel to Game of Thrones. And so thank you so much for your support. Wait, I do have more McGee Lab pictures. I also want to thank Lauren, who's just like such a genuine person and a good friend. And I've also enjoyed just working with you. Like you're a very good colleague to have in addition to a friend. I want to thank my committee, Ruth Ann, Jauji, Andrea, and Mitch, who unfortunately couldn't be here with us today. But Ruth Ann, as the chair of my committee, you made sure that I always felt so supported, and you always bring so many good ideas to the table. Jauji, thank you so much for all of your insight, and you always push me to think about my research in different ways. So I really appreciate that. And then Andrea, as we saw today, you always push me to think about the clinical aspect of things. And as an MD PhD student, that is so important. So thank you. And even though Mitch is not here today, I do want to thank him. He is such a great scientist, but also just such a fun and positive personality. So thank you. I want to thank all of my friends. I see a lot of the MSTPs here today, so thank you for coming. I also want to give a special shout out to Megan, who has been such a good friend throughout this entire thing. We started from the beginning, even during interviews together, and I'm glad that we've stayed friends throughout the whole thing. Joash and Jonathan, I saw Joash here today. Thanks for coming. We have such great dinners and hangout sessions together. And then I want to thank mentors who have become friends as well. So Harper, thank you so much for all of your support throughout the year. He was my high school teacher who I have continued to keep in contact with to this day. And you always know, what's important in life, and we have great conversations, both academic and fun. And then Michelle, as well as my undergrad PI, I would not be here today without you. So thank you so much for your the scientific experiences that I got in your lab. This is just a quick dopamine release of all of my all of my pets. I clearly have a thing for black and white dogs. This is my my family pet Oreo and my personal dog Buka, and I want to give them honorary PhDs in emotional. Okay, so I have such a big family, but I am so thankful that my family has grown throughout the years. I want to thank my partner Keith, who is the best partner that I could ask for, and all of his family who has embraced me into their family as well, especially Karen, Jenny, Mike, and Linda, who are all here today. So thank you. I also have to thank my best friend Nona, who has been my best friend since high school and probably knows me better than I know myself. So thank you so much for all of your support throughout the years. Your sister has become my sister, Jane. Thank you for being here. You are such an inspiration and a fierce friend, so thank you. I also want to thank the rest of the Rosina family, David, Max, Mike, and your parents, even if they're just here with us in memory. I want to thank my siblings. So Tess, who asked great questions about the mice and their experiences. She is such a caring and fun person. Simon and Allison, you guys are just genuinely my friends, and thank you so much for coming today. More sibling pictures. We're awesome. And then Sam and Jane, who are here too, but I think they're kicking it outside because research is boring for them. And then I want to thank my parents. So I was so lucky to be raised by so many wonderful people. So I want to thank my mom, Connie, my grandma, Mary, my Aunt Colleen, and my Uncle Dan. All of you guys have been so instrumental in raising me. So thank you so much. You guys are my biggest supporters, and I cannot thank you enough. And then finally, my amazing grandma had 17 kids, so I have so many aunts and uncles. And I want to thank some of them that are here today. So my Uncle Greg comes out and supports me for all of my different events. My Aunt Michelle and Uncle John as well, who are here. My Aunt Claire and Aunt Shar as well. And yeah, so with that, I will close out.
We'll be meeting for cake and champagne in the reading room at 3 o'clock, assuming all goes well. There is a the thesis committee will, I mean, we noticed that you were very complimentary of the thesis. We will examine her for a few minutes and ask her some hard questions, and we'll see you in the room.
If you're family, and I can show you how to get there if you're not.
Or in the.
Thanks.
It's my work, great option. I won't be able to